Assalamu alaikum, my name is Dr. Syed Waqar Shah and you are watching IGS Biology. The topic of today is DNA replication. What the replication is, why the replication is required. These are the two primary questions. The answer to the first question, when the duplex DNA, when the double-stranded DNA or parental DNA divide into two daughter DNA, the process is called as replication. And the second answer is why it is required. So prior to the cell division or mitosis, a DNA need to be undergo the replicating process. So in the S phase of the cell cycle, the DNA undergo division process. That is, a parent DNA divide and produce two daughter DNA. And when the two copies of DNA develop, the cells undergo mitosis and finally develop two daughter cells. As soon as concerned with the steps of the process of replication, I would like to tell you that it is a continuous process. But for the convenience study, we divide the process into three steps. That is initiation, extension or polymerization, termination phase. Starting with the first one, initiation phase. Initiation phase is characterized by the formation of replicating bubble. Is characterized by the replicating bubble or a replicating fork. Generally, DNA helicase and the gyrase are the two enzymes which got attached with at the replicating site and initiate the process of the replication. What gyrase do? Gyrase open turns of DNA duplex by breaking the base pair. The gyrase open turns of the DNA duplex by breaking the base pair. However, the helicase facilitate the unwinding process. Helicase facilitate the unwinding process of DNA duplex. So, with the action of these two enzymes, the DNA fragment start open like a zipper. And it give a bubble-like appearance. And this is called as replicating bubble. Beside it, each side of the replicating bubble is called replicating fork. When the replicating fork develop, an SSB proteins got attached with, and this SSB protein is pronounced as a single-stranded binding proteins. And these proteins never allow the two strands to meet again or to join again. Once the replication bubble or replication fork develop, the process of initiation completed. And after the initiation, the second step, the second process started, that is extension or polymerization. In the process of extension, the two enzymes are involved, the primase and the DNA polymerase. These are the prime enzyme, or these are the most important enzyme which help in the extension process of the DNA. So the primase, what the primase do, that the primase develop a primer, which is 10 to 12 RNA sequence. It develop a primer, and the DNA polymerase work according to the primer, which is got attached at the leading strand or the lagging strand. The primosome, the primase and the DNA helicase, when work in a complex, it is called as primosome. So the term primosome means when the primase enzyme and the helicase enzyme work in a complex, it is called as primosomes. So before going into the extension process, we have to see how many types of DNA polymerase are? So there are three types of polymerase. That is polymerase 1, DNA polymerase 2, and the DNA polymerase 3. DNA polymerase 1 replaces the RNA primer by DNA nucleotide. So the work of the P1 is to replace RNA primer by DNA nucleotides. P2 repair and remove the error of replication, repairing process and to remove the error of replication. And DNA polymerase 3 is the main enzyme which synthesizes daughter strands. So when the replication bubble develop, there are two type of strands develop. One is lagging strand and one is the leading strand. How can we identify these strands? We can identify these strands by the direction of the replication. If the replication occurs toward the fork, this will be the leading strand. And when it occurs away from the fork, this will be the lagging strand. The process of the replication along the leading and the lagging is slightly different. Although they have the same direction, that is occurs from 5' to the 3' direction. But the process is different. How it is different? That along the leading strand, the process of replication is continuous. However, along the lagging strand, the process of the replication is discontinuous. Along the leading strand, what happens that the primase enzyme attach a primer at the site of the replication. When the primer attached, the DNA polymerase 3 got attached here and add the nucleotide according to the primer, and that's how the leading process that is replication process occurs along the leading strand. Later, the axonuclease enzyme remove the RNA sequences and add the DNA sequences. And ligase enzyme finally strengthen these bases along the leading strand. At the lagging strand, what happens that addition of Okazaki fragments occurs discontinuously along the lagging strand. Okazaki was a Japanese scientist who introduced these fragments. That's why they are known as Okazaki fragments. During the addition of these strands, polymerase repair and also the process of removing the nucleotide errors done by this polymerase 3 enzymes. So the lagging strand completed. When these two strands replicated, they got separate, and finally what we have a two DNA strands. Here exonucleases done the same process, that it removed the RNA sequence and add the DNA nucleotide, and the ligase enzyme strengthen the new nucleotides along the length of the Okazaki length of the lagging strand. So when the process completed, the final process occurred, which is called as termination phase. After the replication, DNA ligase, I have told yourself, and they will develop when it strengthened the nucleotide along the length of the leading and lagging strands, the daughter strands got separated. When they are separated as an individual DNA, the process of termination occurs. This is called termination phase. This process of DNA is called semi-conservative because it occurs along the template DNA, and the template DNA is the old DNA, and the new strand develop along the existing old DNA. This is called replicating process. So, want to give you a quick review of this process. That the replication is a process in which a parent DNA undergo division and produce two daughter DNA in the S phase. It is a continuous process. For our convenience, we divide it into three steps, that is initiation, extension, polymerization, and or termination process. Initiation is characterized by the replicating bubble or replicating fork, and this process initiated by two enzymes, gyrase and the helicase. Gyrase open turns of the DNA duplex by breaking the base pair, while the helicase unwinding the DNA duplex. This action of the two enzymes open the DNA like a zipper, and that's how a structure develops, which is called as replicating bubble. And each strand at the replicating bubble is called replicating fork. When the process of this process completed, the SSB proteins got attached with and keep these strands separate. In the process of extension, leading and lagging strand develop. Along the leading strand, replication occurs toward the fork, and along the lagging fragment occurs away from the fork. In the leading strand, the primer got attached by the primase enzyme, and according to the primer, new nucleotide added by the P3. And this P3, when completed the process, exonucleases remove the RNA sequence and add the DNA nucleotide, while the ligase strengthen these nucleotides along the length of the leading strand. However, at the lagging strand, Okazaki fragments attach discontinuously along the length. Similarly, here the exonucleases done the same process and the ligase done the same process, a strengthening of the nucleotide along the length. When the replication completed, the two strands got separated from each other. When they got separated, this process is called as termination. And when the termination occurs, we have two copies of DNA. This is called as replication. So this is the overview of replication. I hope you people understand it well. If you have any question, you can ask in the feedback section. Keep watching IJS. Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh.